Polymerase chain reaction, or PCR, is a technique that is used to amplify a DNA region that is flanked by two synthetically made primers. The first step in PCR is to identify the region found in a template DNA molecule that you wish to amplify. If the sequences at each end of this region are known, primers can be generated that are complementary to these two sequences. Since DNA molecules consist of two antiparallel strands, the primers can be designed so that the primer hybridizing or annealing with the one strand will have its 3' end toward the center of the sequence to be amplified, and the newly made strand will be made in that direction. This is considered a reverse primer, while the primer annealing with the other strand will have its 3' end also toward the center of the sequence to be amplified, and the newly made strand will be made in the opposite direction as the one made on the other strand. The primer that anneals with this strand is considered a forward primer. The PCR reaction would also contain deoxyribonucleoside triphosphates of adenine, guanine, cytosine, and thymine, and thermostable Taq DNA polymerase. All of these components are mixed together in a test tube and placed in a thermocycler. This machine has the capacity to rapidly change temperature from a strand separating temperature, usually around 90 degrees centigrade. Once the strands have separated, the thermocycler temperature progresses to a primer annealing temperature that is dependent on the specific sequence of the primers used, but is usually between 50 and 60 degrees centigrade. After the primers anneal, Taq polymerase synthesizes complementary DNA strands by incorporating deoxyribonucleotides at the 3' end of each primer, using strand A or strand B as the template. This is referred to as one thermocycle. In order to amplify a sequence, the sample is usually taken through 30 to 40 thermocycles. When the first thermocycle has been completed, a newly formed strand 1A and 1B will have been formed. During the second thermocycle, the same process is repeated, generating 4 new DNA strands. At the close of the second thermocycle, we now have 4 DNA molecules with 8 strands. All 4 of these molecules contain the sequence we wish to PCR amplify, but none of the 4 molecules we have made thus far exactly end at both ends with the primers. That is accomplished after the third thermocycle. With each subsequent thermocycle, the relative number of these molecules increases until after many cycles these will be the predominant type of molecule resulting from the PCR.